So this is part two of chapter two, taking a look at, part two of chapter 12, taking a look at neuroglia. You might also find, if you look up these types of cells in other sources, that they're also referred to as glial cells or just glia for short. But these are cells that are not like neurons, but they are supportive cells for neurons. So we'll get into some general characteristics about them and then some more specific ones. So glial cells or neuroglia, these do not function the same way our neurons do. They're not passing signals so that we can maintain homeostasis in the body. They're non-excitable, but they do provide support for those excitable neurons that we see and that we use to live. Um, there's probably about the same number of glial cells as there is neurons, but as we age and if certain things like neurological disorders may set in for certain people, depending on what happened to them during their lifetime, glial cells could outnumber these neurons. A few characteristics of these glial cells: they are different from neurons in that they are able to do mitosis. So if some of them become damaged or old, they are able to do mitosis and do cell division and replace themselves. We know that this is not a characteristic of neurons. Another characteristic is that they protect and nourish neurons. They do this in different ways, and it depends on what type of neuroglia it is. So we'll talk about that. Certain types of neuroglia provide physical scaffolding for nervous tissue. What this kind of means is a place for those neurons to migrate to when they are forming our different structures, like the brain, spinal cord, and all those nerves. So this happens during development in the womb, but it can also happen with using physical scaffolding if we have an injury to our nerves. This is specific for the PNS. And then, of course, these types of cells, since they are protective and nourishing for neurons, they are critical for neurons to function normally at their synapses where they connect with each other. So we'll start with glial cells in the CNS for this one. So astrocytes are one of those cells. Astrocytes, they are star-shaped cells, that astro meaning star, and they have what we call these perivascular feet that are going to actually help these cells connect neurons to a blood supply, which is why we call them forming the blood-brain barrier. They are going to control what different substances come to the neurons from the blood, which means that they regulate tissue fluid composition. So a lot of times they're just regulating ion levels that make their way to the neuron. We care about ion levels because this is how we're going to actually form synapses and change electrical membrane potential, right? Based on what ions go in and out of the cell. They can also provide some structural support. They assist in development. They alter synaptic activity. So since they are a connection for these neurons to a blood supply, to different ion levels, they can affect activity. And in case we have damage to those neurons and they die, usually astrocytes will do mitosis and take their place. I'll scoot over to that slide real quick to show you a picture of this. This green cell here is an astrocyte. You can see it's using its perivascular feet, peri meaning next to, vascular meaning blood vessel, to connect this neuron here. We have a multipolar neuron connected to a blood supply here. Astrocyte. The next type of cell is called an ependymal cell, or ependyma for a cluster of them. These are found also in the CNS, so the brain and spinal cord. They make up part of a large group of cells, which we call choroid plexus. And choroid plexus is a group of cells that actually produce cerebral spinal fluid, or CSF. These ependymal cells, they are going to regulate what concentration of certain ions from CSF are able to make it to neurons. So they're doing a very similar function to astrocytes, except they are manipulating CSF instead of blood. We can see these ependymal cells here. They are lining spaces in the brain and spinal cord, and we can tell we're in the brain here, not just by this picture, but because we can also see that these cells are lining a ventricle, which is an open space in the brain that fills with cerebral spinal fluid. For our other cells in the CNS, one is microglia. These are not fixed cells in the brain or spinal cord. They actually migrate to different areas where there might be tissue damage or a pathogen present. And they are immune cells. They are phagocytic, meaning that they are going to engulf anything that shouldn't be there and try to remove it. And I'll show you that's these kind of teal colored cells throughout here. And then lastly, we have oligodendrocytes. These cells are not directly on top of the axon of CNS neurons like we might see with the PNS neurons, but they do create these myelin sheets over the axons of the neurons. We're going to look at the function of myelin today, but this is going to help with insulation and passing of the electrical signal across the axon very efficiently and quickly. So oligodendrocytes are the ones providing this myelin sheath to the axon of neurons. You can see them here, not directly associated with the neuron, but are producing these myelin sheaths for the neuron. You can also see they're providing some scaffolding here, kind of connecting neurons together and letting them kind of live together with some structure. If we go to the PNS, so the peripheral nervous system, where we have our nerves, there are two different types of cells. One of them is satellite cells. If you remember, when we were looking at the PNS, it's separated into ganglia and our nerves. Nerves are just bundles of axons versus ganglia, which are the cell bodies of those neurons. And so these satellite cells help keep the cell bodies of the neurons together in the ganglia. They also can provide some insulation as well as regulate potentially some of the nutrients and waste that are coming and going from these neurons in the ganglion. The other type of cell, which is very important, are Schwann cells, also called neurolemmacytes. These are actually attached to the PNS axons, okay, and they are providing myelin so that we can do faster action potential propagation. Very similar or same function as these oligodendrocytes, but in the PNS, they are actually directly associated with the axon of those neurons, right? You can see the nuclei of these cells, and they're just adding layers and layers of myelin. Again, these are called Schwann cells or neurolemmacytes, and they are exclusive to the PNS. Here's the picture for the satellite cells, a bunch of these small pink cells kind of surrounding and insulating, arranging what nutrients can come and go to these cell bodies in the ganglia. So let's talk about myelination, since we mentioned two neuroglia that are providing myelin sheaths to the axons of either CNS neurons or PNS neurons. I will say myelination is necessary for these axons to function efficiently for sending signals between cells to their effectors. And so myelination is the process where we have an axon from a neuron. It could be from the CNS or PNS, and myelin is being added onto that axon, and that is through the action of specific cells. So again, in the CNS, oligodendrocytes are doing this. CNS meaning the brain and spinal cord, so whatever neurons are there, oligodendrocytes are what are going to be adding that myelin sheath. And then in the PNS, it's going to be those neurolemmacytes, also called Schwann cells. You can learn either one. Either one is acceptable on an exam. Myelin itself is lipid-based, and it provides really good insulation for the axon, especially if electrical signals are passing through the axon. We'll give an example here in the PNS. So a Schwann cell or neurolemmacyte is going to encircle, as we can see in this picture, because they're directly associated with the axon of the neuron, and they wrap around it and they start forming these sheets of myelin. And kind of when we take a look at this, the neurolemma's cytoplasm, its nucleus, they're all kind of pushed to the outside surrounding those layers, and these cells collectively we would call neurolemma. Neurolemma will be really important when we talk about the ability to do regeneration and guiding axons back together when they've been severed due to injury. And typically, neurolemmacytes are going to myelinate a specific portion of the axon, and they won't do all of it. We can see in that previous picture, you can see there's gaps here. So they're only going to do about a meter long, okay? And then there's these gaps, a millimeter, excuse me. There's these gaps, and those gaps are called neurofibril nodes or nodes of Ranvier, where there's just spaces on the axon, as you can see, that are not myelinated. Oh, great. We have a good picture for that as well. Okay, so we've got our neurolemma sites. That surrounding area where there's the cell and the cytoplasm is called the neurolemma, and then it's making these sheets of myelin that are wrapping around the cell. In the CNS, something very similar, right? Okay, we've got these cells on the outside though. So instead of us forming a neurolemma on the outside, which is connecting these cells directly onto the axon, we don't have a neurolemma here in the CNS because these oligodendrocytes are kind of outside all of it, just projecting and putting myelin sheath onto here. We still have those nodes of Ranvier, also called neurofibril nodes, okay, where there's spaces where there is not myelin present. We do have a few clinical views. Again, these are going to be fodder for essay questions on an exam. One of them is multiple sclerosis, also called MS. Okay, a lot of us may have heard of this or know someone, a family member or a family friend who has had this. This is multiple sclerosis. Typically means multiple as in many, sclerosis meaning scars. Okay, so many scars. And where those scars are happening are going to be on the axons of neurons because multiple sclerosis describes the progressive demyelination of neurons in the CNS, meaning that these lovely neurons, I don't know if I have a picture, I do not, meaning these lovely neurons, okay, are going to have demyelination occurring where these sheets are going to be broken down and it actually leaves behind scar tissue, which makes the neurons not very usable, okay? They can't do their purpose. Multiple sclerosis is an autoimmune disorder where the oligodendrocytes are attacked by immune cells, so they are no longer producing that myelin sheath, which insulates the axon of those neurons. And again, if we don't have myelin insulating the axon of neurons, we're not going to be able to deliver signals very efficiently between each other, okay, between these neurons. Repeatedly, this causes inflammation, which causes scarring, and then we may lose function completely of those neurons, okay? So this is a progressive disorder. Another clinical view is Guillain-Barré syndrome, okay? Loss of myelin from peripheral nerves due to inflammation. So this is affecting the PNS. Typically, these are going to be nerves that are going to effectors like muscles, and it is described by muscle weakness, especially in distal limbs, okay? And typically gets worse. So it starts in those distal limbs, arms and legs, and can get worse as we get closer with different muscles. This is progressive as well. A lot of times function can be recovered, okay, with some medical intervention, mostly just taking care of this inflammation so that we can use those peripheral nerves again. With multiple sclerosis, this is a little bit more difficult because once you've kind of lost myelination of those CNS axons, that scar tissue and those neurons themselves do not do very well, and the likelihood of regeneration is very low. Kind of a last clinical view, and this has to do with regeneration possibilities. I really do want to note, I think I kept that slide. Yeah, okay. I do want to note that regeneration of an axon in the CNS is not very possible, okay? But in the PNS it is. So if there's a traumatic injury to PNS axons, which we know are nerves, they have the possibility to regenerate. They can regenerate if the neuron cell body present in those ganglia is still intact and if enough of the neurolemma remains. Remember, neurolemma is the body, nucleus, cytoplasm of those Schwann cells, neurolemmacytes. Again, this is possible, right? If we have enough of these neurolemma and if the cell body of the neuron itself is intact, even if its axon has been damaged. More likely if the amount of damage is not very extensive, right? And it really depends on the distance between the site of the damage and the structure it innervates. If that distance is shorter, much more likely that we will have regeneration occur. If it's over a much longer space in the body, maybe not. These are some of the steps that happen. So the axon is severed by trauma, okay, as part of the nerve. Um, Typically, that axon, where it is cut, okay, damaged, will seal off, okay, and swell. And then what we have is part of these neurolemma end up surviving, okay? We might have some degeneration of the axon, okay, which is called Wallerian degeneration. So where that axon was cut, whatever's been cut off from it will likely degenerate. But if that neurolemma survives, it will start to form what we call a regeneration tube, and it will guide certain elements needed to help us regenerate that axon that was degenerated using these neurolemma sites. So these cells are going to help us guide different factors that are going to help us grow the axon back. The axon will have reinnervation and will be able to do its normal function. That is usually what happens. This is exclusively in PNS axons if we have damage there. Again, it is dependent on a number of factors. For the CNS, it's extremely limited. Oligodendrocytes can secrete certain things, okay? But they're not going to be growth factors to help us regenerate any degenerated axons, okay? Um, and what we see is oftentimes if we do have damage to these axons, they end up scarring, and then regrowth is pretty much obstructed from these scars as well as from other cells like astrocytes and other connective tissue coming in and taking the place of those damaged neurons. So if we have damage to the CNS in the brain or spinal cord, likelihood of axon regeneration is going to be really limited. We'll get to part 3, okay, communication with these neurons, and there's going to be a lot of steps to this, so hang tight for chapter 12, part 3.